So as already mentioned, I will talk about analysis of polymers, and I have divided it in a way that all of the examples are related to something that we have worked on in my team. And then from these examples, I will share my own experience of some of the situations where I had a very steep learning curve to come out and solve some of the problems that I encountered on the way. So this will also be like my own recap of problem-solving projects. And first of all, as we will be talking about polymers, and I'm not sure which exact background each of you has who's attending the seminar, I will give a very brief introduction to polymers as a chain of many interlinked monomeric repeating units. So we have monomers, we have them linked together in a very long chain. So we have many individual fragments being put together. And if you have a look at both natural and synthetic, organic and inorganic elements, then we find, well-known DNA, RNA, cellulose, like many biopolymers. We also find diamond and graphite as inorganic materials with many repetitive units. And if we go for the synthetic one, then we have the polysiloxanes in the inorganic, but where I will focus on today, many functional materials that you know and that have been a large part of everyday life, and nowadays also of many of the environmental problems associated with them that we need to overcome in the future. So we have PET, nylon, polyesters, and many more of similar materials. And one of the most important parts of these polymers when we are discussing and analyzing them is that they are not as uniform as we make them look like. So if I show you a drawing of, let's say, 20 or 30 repeat units, that doesn't mean that every single strand inside of a sample contains exactly this number of repeating units. And here you see a multi-analysis of a polymeric sample, and you can see here you have masses between 2,400 until 5,400. And each of these peaks in the mass spectrum corresponds to one difference in the repeating unit. So this means we have a very broad distribution of different chain lengths, which will be a major impact, will have a major impact on our analysis of these materials. And then if we first have the connection of the individual parts, then we also have to consider the composition. So far, I've only shown you a so-called homopolymer, so one polymer strand which consists of repeating units of the same kind. But of course, we can have also two or more different types of monomers that will then copolymerize. And if you look at the copolymers, then we can have strictly alternating monomeric units. We can have a random distribution of these units, and we can also have block copolymers, so block distributions of the individual monomers. And of course, this also has influences on their properties. During what I will present today, I will mostly focus on these blocks of polymers, but be aware that there are lots of researchers also working with the other different types of assemblies. And then if we talk about the connectivity, which was the previous example, we also have to include what this results into. So in the previous composition example, I had mainly linear chains. But you could also think that the materials can be branched, or they can be cross-linked, both physically and chemically, to form, for example, cross-linked polymer networks. And this then, of course, also has, again, influences on the properties on the materials resulting from it. And again, we can have further examples. So I'm only giving examples. This is not a conclusive summary of all the existing ones. But if we think of different polymers, also polymers consisting of more than one monomer, then we also have to consider self-assembly processes of these materials, in particular if they are different in their hydrophilicity or how hydrophobic or polar they are. And we can have structures of these self-assembling materials forming micelles, nanogels, where we have interlinked micelles. We can have worms, vesicles, so a very rich structural diversity in the 3D space as well. And of course, copolymers can also form these cross-linked chemical structures. So these ones where we are looking at all the different repeating units as just blobs. But of course, as chemists, we have to fill them with chemistry life. So I've tried this for two different examples where there's not just the connectivity, but also the stereochemistry that we need to take into consideration. And the left example deals with polypropylene. So from the first glimpse and from the chemistry background, we would say it's a rather straightforward example or a straightforward molecule. However, if we have a look at this methyl group, then this methyl group can have a different arrangement with respect to the overall molecule, and we can have all the methyl groups pointing to the front or in the same direction. Being an isotactic case, we can have a syndiotactic alternating arrangement in 3D of these moieties, or we can have an atactic where all the functional groups or all these methyl groups are pointing to random sites. And this is not just the case if we have propylenes or alkyne chains, but for example, we also have to consider this if we look at polypeptides and polypeptoids. So if we have these amide functional groups, then they have partial double bond character, which is indicated here by these dotted lines. And this means that for these type of structures, we have to consider also cis arrangements and trans arrangements with respect to this partial double bond character. And all this stereochemistry has also an influence on the properties. So crystallinity, for example, depending on how well these individual structures can pack or get ordered. But of course also, and this is related to what we are doing with NMR, there's extensive work on the influence of the tacticity on the chemical shift. And there's lots of work on how different triads, so if you pick out 3 units, how this influences the chemical shift that you will later observe in the spectrum. So this is a very important part of investigating polymers. So now, as I promised beforehand, I want to dive into 4 different example projects. And I will go from solution projects to gels and then to completely solid. So I will span across all the different aggregation states or states of matter that you can have in that respect. So first, I would like to introduce you to analyze polymers in biorelevant environments. And in particular, I would like to introduce you to these colleagues here, which are called FASIF and FASIF, which is the abbreviation for fasted and fed state simulated intestinal fluids. And this means that these 2 mixtures that you can buy from the company biorelevant can be used to simulate the fluids in our intestines. And this makes it very attractive because we can use in vitro tools to predict already behavior in vivo. And we can, for example, use this to get information on how well different drugs, for example, would dissolve in the intestinal fluids and how additional components, excipients, polymers, etc., would influence this dissolution process and would then later also influence bioavailability. So here's a summary of how these biorelevant media look like or what they are composed of. So we have the important taurocolate, which is the amphiphilic molecule in this mixture, and this is necessary to be able to solubilize all these long chain alkyl components. So these would all be fatty acids and derivatives that are not soluble in water in sufficient quantities, but together with these taurocolate molecules at a defined pH, they can form a really rich colloidal chemistry. So this is part of a video that you're welcome to watch from our pharmacy department here at Würzburg, and you can see that in the stimulating media, but this means also in our intestinal fluids. So right now in your gut, you have micelles, vesicles, rod-like particles. So you have a lot going on, let's say an aggregation party going on in our intestinal fluid. And having this now prepared in D2O, it means I can throw polymers at different concentrations into the mixtures. And then I can observe for commercially one, you can see here the very famous class of fluoronics, or for one that we got from a collaboration partner made up of polyamides. You can see that if we increase the concentration of this commercially available polymer, we also see an increase in the change in chemical shifts for all the components in the mixture, while for the second polymer, there's barely anything happening if I increase the concentration of the polymer. And this might be a first indication, a hypothesis, saying, okay, the first one is introduced into the colloidal party or self-assembled party, while the second one is like bystanding and is not massively influencing the composition and the colloids that are forming. But ideally, I would like to analyze this in more detail. So if you think what could be an interesting tool, then we are coming towards diffusion NMR, because this would allow us to track the different components and to track in which aggregates they are present and how this aggregation state is influenced upon addition of other compounds. And if we do a small recap, then DOSY is really cool and powerful because we can trace how molecules move. So by using gradients, we can spatially encode molecules. Then they have time so that they can move somewhere else, and then we can decode their position with a second gradient. And we are doing this by checking out how the signal intensity changes. So if a molecule A just diffuses a bit, then the signal will decay also a bit, because at the end stage, the magnetic field that this particle will experience will be different from, for example, B, which moves a lot faster, and then the signal will have decayed a lot more. So for each of the individual and distinguishable parts of our molecules, we can get these decay curves, so the relative intensity of our signal compared to the gradient, or as a function of the gradient strength. And we can plot this in a series of different ways, all making use of this equation for the intensity. So this would be what we would normally do for a small molecule. And for a small molecule, we have nicely separated signals, and we also have a nice decay that we can fit with a mono-exponential function. If we now go to polymers, then it's getting a bit more complicated. So here you can see a work where Burkhard Louis was involved, and this is now diffusion NMR on polymers. So you can see here the different spectra, where with increasing gradient strength, you have the decay of the signal. And then, of course, for each of the signals, you can plot the signal intensity as a function of the gradient strength. And then you have to fit this. And here they have shown the fitting procedure using different mono-exponential decay functions, but they have also tried two models, the gamma and the log normal function, to account for the fact that the polymer is indeed not a monodisperse, but a polydisperse sample. Remember the distribution of the different molecular weights. And this is taking them into consideration, are we able to fit the experimental data? And in this work, they have showed that they are able to get molecular weight distributions by pulse field gradient NMR that are in agreement with those that are obtained by the more established method of size exclusion chromatography. And this is not the only work that is related to DOSY on polymers. So I have a small collection here of other works, for example, here where DOSY was used to facilitate the molecular weight determination. Then there's one where there was even a specific probe design to make this more efficient, this determination. And then there's also a quite recent work where different approaches and setups are compared to be able to, in the end, choose the most appropriate set of parameters and pulse programs, etc., to characterize these polymers and extract molecular weights. And for those of you who also prefer a more tutorial or webinar based, there's also a webinar on demand by Brooker that you can also extend the information on this type of analysis. Okay, so this means we can now have a look at the materials that we have here at hand, and I want to show you some exemplary attenuation curves that I've plotted here for passive. So this is the individual fed state simulated fluid, so no polymer yet. And you can see that down to a signal of just 1% intensity of the initial one, this is, and even a bit lower, this is very well to fit with the mono-exponential function. Then as soon as we add the polymer that you see here on the top, then you can see it's still kind of okay, but it's getting worse. And as soon as we add polymer and the drug molecule that you see here, you can see that this is far away from this mono-exponential fit that was applied for the left two cases. And the fact that in the middle situation, it's still kind of okay, stems from the fact that this polymer that I've shown you here in the analysis has a relatively small distribution of environments. So this is with a small polydispersity index. And this is why I've also brought you one example from one of my PhD students who has worked with a polymer based on hyaluronic acid. And this is from a specific diffusion probe. And you can see that at this 1%, we are far away from being able to fit this with a mono-exponential fit. But here you can see that we can reproduce the data by using this log normal distribution from the publication that I've shown you 2 slides before. And by taking this into consideration, the dispersed nature of the polymers, it is then possible to also distinguish the individual components. So I've shown you here one example from an extensive study that we've done in FASIF. So on the left side, you see the example only the FASIF with the polymer. And you can see here on the left side, the tarocholate in black, which is the one that is soluble, so it diffuses fast. And then we have the red, which are the lipids, and the blue, which are the polymers, which diffuse slower. And they diffuse with the same speed. So we can conclude that they must form some kind of mixed aggregates. And then you can also see that as soon as we increase the amount of polymer, these aggregates also start to diffuse slower and slower, with a hypothesis that they form larger species of lipids and polymers together. And this would also be in agreement with this proton NMR data that I've shown to you. And what was even more interesting was as soon as we add a drug molecule, this is the green dots on the right side, you can see that it's even more diverse. So the green molecules diffuse first with the lipids. The polymer is doing its own stuff. At a certain concentration, this all collapses, and from there on, they diffuse together. So as a result, for the other polymer, we can do a similar behavior. And the result here from this study was that we have a concentration-dependent behavior, as you can see by these developments, but that we also have a composition-dependent behavior. And that, for example, if you think of this passive media as a tool to screen polymers, then just screening all polymer materials by themselves would not be sufficient to account for the behavior in the intestinal fluids in the presence of, for example, drug molecules. So this leaves me to go to the next example. And this is the thing with the relaxation. So I've promised you these block copolymers, and I would like to briefly focus on these tri-block copolymers that have in blue hydrophilic parts, in red hydrophobic parts, and again hydrophilic. And if they are dissolved in water and they surpass a certain critical micelle concentration, CMC, then they self-assemble into these core shell structures, and they are in the area of several nanometers. And in the theory, we have the shell, which is water soluble and in contact with the water, protecting the particle and stabilizing it, while in the center, there's the more hydrophobic core. And this is also where ideally cargo could be transported, or low water soluble molecules might be stored. So this is the ideal image that we have of these particles. And when I started to work on them, I got some samples from a collaboration partner, and we said, okay, micelles is a phenomenon in solution. If somebody needs to take medicines, will probably also be in solution. So let's make analysis in solution. So here you see the proton NMR data in chloroform of polymers with the drug evavirons that you've seen already on the previous slide. And if I analyze the same sample with the same amount of evavirons in water solution, in aqueous solution, you can see that here in the aromatic region, where there should be signals from the drug, there's nothing to be seen. So essentially, we are invisible to what is happening in the core of this particle. And this is not just the case for this one example of a drug molecule, but here's the natural product curcumin, also encapsulated in such a micelle. you see here the equivalent proton spectrum, and if you really zoom in, you can see that there is something in the aromatic region that is incredibly broad. And if you increase the temperature, you can also make this sharper as you increase the mobility of this particle overall. And the reason why we have this problem is based on the relaxation. So if we briefly recall that in small molecules, we have a very fast motion and we have a more or less okay, but not very efficient relaxation, both T1 and T2. And then as we go to larger molecules that are also moving slower, so-called intermediate motion, we have the minimum of the T1. So this is where T1 is fastest and T2 is also getting faster. And then after reaching this minimum point, the T1 is again getting less efficient, while the T2 is getting more and more efficient with increasing molecular size or decreasing motion. And then I don't need to remind you that with polymers, super large molecules, or with solids, we're here on the right side and kind of getting the worst from both worlds. We're having long T1 times, which means long experiment times, and we have short T2 times, which means that we have broad signals. And I would like to introduce you to two examples covering this T1 and T2 part. And for the T1, I would like to focus on these dendrimers and work by Dahlwächter and Reichert that was published as a book chapter. And you find these branch dendrimers and different generations, so different sizes of the overall dendrimer. And what you can see for all of the different dendrimer sizes is that the terminal sites, so the outer functional groups of this dendrimer, they have a variation in the T1 time, which is also slower than the internal sites, and these internal sites have a constant one, independent of the size of this dendrimer. And this also then has consequences. For example, if you want to quantify the functional groups, and even more so, if you have a polymer which can have even more repeating units than you have generations in this dendrimer. So you have to be very careful when you set up parameters for the experiments. And in particular, it has consequences when you are doing determination of the number of repeating units, because it's very common to use the end group analysis, where you have one single end group that might be different in the mobility than the rest of the polymer chain. Plus you have a lot of repeating units for the polymer chain, which also makes the error quite large when you determine the number of repeating units. So here you have to be very careful with the setup of the parameters. And then with T2, I have brought you one example from a textbook, from the Claridge textbook, where the short T2 time of polystyrene, a very large polystyrene polymer, which gives very broad resonances in the NMR spectrum, have been suppressed using, for example, a CPMG sequence, which causes T2 editing. So everything with a short T2 is vanishing from the experimental spectrum. And then you can see here that you can use it, for example, to distinguish between different components. And this I will also come back to later, because I also have an example for making use of some of these filtering experiments for solids. So this means that on the one hand, we can have disadvantages from these properties, but we can also use changes in the relaxation behavior to our advantage, or to learn something about our system. And here I've brought you one example of a systematic study of, again, a similar polymer of the ones I've shown you before, that was loaded with different amounts of curcumin, the natural product. And you can see here all the signals being assigned. And then as you go and increase the amount of curcumin that is entered, you can see that the signals broaden or lose in intensity. And then, for example, you can look at specific ones. So here would be the green ones, would look at the change between the hydrophilic polymer segments and the hydrophobic polymer segments. And then you can see, for example, that this ratio increases drastically as you go to higher loadings. And you can also make this for other functional groups and then use it complementary with lots of other tools. Almost none of these examples is able to increase our understanding of the materials sufficiently just by using one technique, but in all of them, they are complemented with lots of sophisticated analytical tools to get a better overall understanding. And in this case, the resulting picture or idea that we got from the studies also at high loadings, there's kind of a jelly formed so that some of the material is also precipitating. And we could, for example, observe for 3 different polymers and different amounts of drug or gas molecule inside that we have states in which all the signals are nicely present, where everything in the particle is well hydrated. Then we have an intermediate region where this is starting to suffer. And then we have the moment where we have these jelly agglomerates that are forming so that particles are sticking together when we see a large drop in the signal intensity. And this is some of the ideas that have been derived from taking a closer look at when we are losing signal intensity. And this is a very good jumping point, because from this having something jelly and losing intensity, I would like to jump to hydrogels, and in particular hydrogels that have been used for 3D printing and for tissue engineering. And hydrogels you have of course also come across in your daily life if you're using contact lenses, in diapers, in 3D printing and lots of other materials. There's hydrogels present. And I've brought you one example of 3 different polymers. I will only explain the first because the other ones are just derivative to get additional structural information. So we have again these polyamides, and we have in the first part a hydrophilic block. Then we have a phenyl aromatic ring containing hydrophobic block and another hydrophilic block. And for all of these triblock copolymers, if we dissolve them in 20 weight% in water, then we can observe that at low temperature, they form hydrogels. And you can see here in the image, these worm-like structures. And when you heat this above roughly 30 degrees, then these worm-like structures are broken up and we have the individual micelles present in our samples. And here the idea was, if it is possible by studying this transition process, if we can learn something about the mechanism behind distillation, because these functional units that we have here, they have only hydrogen bond acceptors, but nothing that can donate hydrogen bonds. This means that we need to find other proximities or interactions that are responsible for this sphere to warm transfer. And the first thing that we started with is we thought, okay, let's do NMR in solution and let's cover different temperatures. So here you only see 2 individual examples in the gel state and at the maximum temperature that we recorded. But you can see that we observe changes in the signal intensity. We have overall broad signals as we would expect from a hydrogel or also a micellar sample. Plus, we could also estimate the changes by calculating P ratios for this particular system in steps of 5 degrees. And then we thought, okay, if we found that there are changes in the spectrum, let's see if, for example, by using a tool like DOSY, we would be able to see a specific proximity of functional groups to other functional groups. And then we recorded 2D noisy data that looks quite noisy, but still interpretable in the gel and also in the sol state, so for the gel and for the micelles. And then we looked at the different environments and saw, okay, where are their proximities? And this was a moment where we went to a national German NMR conference and we were asked, like, how many spins are you actually looking at if you're doing NMR in solution? And this was rather a steep learning curve and very interesting to observe. And we were very lucky that we had Kai Saalwächter supporting us in this journey, because he invited us to Halle and measured relaxation times with us. So at low field, so we used his low field NMR equipment and we together did then the data analysis, and we recorded the T2 relaxation times, and were then deciding that the best way to fit this data, excluding the bulk water that we have in our system, was to divide into 3 different regions of increasing mobility. So we had one part of the spins being very rigid, one part being at an intermediate regime, and then we defined one other component that we would associate with rather mobile species. And we could then plot this for each of the polymers, and I would again like to focus here only on the polymer A and on the hydrogel state. So the good thing that we found was that the mobilities and these regimes were in agreement with the gel strength that we got from rheology. So we could already correlate what we found with the low field NMR with other methods that have nothing to do with NMR. And also there were agreements between this and molecular dynamic simulations, which was encouraging. But what was rather tough was, if you look at this part here, so the light gray one, which is all the mobile species, you can see that we are here at roughly 50%. So this means that in the gel state, we only observe roughly 50% of the spins if we are using standard solution NMR. So half of our sample we are blind to because it has decayed already before we do the proper measurement. And this was a very good point and also a turning point, because this was when we also did solid state NMR experiments, so that we probed all the very mobile and then the very rigid parts. And in solid state NMR, we could indeed find indications for additional proximities between some of the hydrophilic and the hydrophobic groups, which are most clearly visible for the polymer that is also the stiffest or forms the strongest gel. And this leaves me to the last part, and this would be to go to the complete solid material. And here it's very exciting if we have a drug embedded in a polymeric material. Using solid state NMR in particular, when you remember that in solution we are blind to some of the spins, for example, the core of these particles, we are doing ongoing work on explaining bulk properties, in this case, for example, the dissolution rate, by what is happening on the molecular level once we increase loading. And here, for example, we could explain, based on where the molecules are located, why it has such a strong impact on the dissolution rate. And there's a lot of interesting work on correlating the structure of materials with the properties, so these structure property relations. And I could name many, but I would like to focus on one that was kind of my inspiration to go deeper into this whole field, which was a work by Martina Stenzel, which she published in 2017, based on a polymer with carbohydrate sugar units. And she was incorporating a platinum drug inside of the micelles that this polymer can also form, but completely attaching the drug to it. And then she studied how well, at different amounts of drug loading, these formed micelles are taken up by cells in a biological setting. And she found that a lower loading resulted in a beneficial uptake compared to a higher loading. And she used solid state NMR, among other techniques, for example, scattering techniques, to explain or derive a structural model that you see here in the bottom, where she made a hypothesis based on the data that in case of the high loading, we have a hard corona, which is not so well hydrated. And this hard particle is much less taken up, and the soft hydrophilic corona, so a softer particle that can be deformed more easily, is kind of beneficial for the uptake. So she correlated things she saw in the NMR data with things she observed in a biology lab. So we started our journey in this respect, saying that we're interested in the cancer drug paclitaxel, and there are again 2 of these polymer systems based on a previous work where the researchers found a polymer that can encapsulate a massive amount of 58% of this drug, so would be super high loaded, very efficient. And this means they could provide us with a series of different amounts of drug loading, and they already had images confirming how these particles look like. The problem with this setting was that we have amorphous samples in this case. So these drug polymer formulations are completely X-ray amorphous. And we also, due to this amorphicity, having a distribution of environments and again, very broad signals, and the complexity of the molecule meant that we had a lot of signal overlap. And now we have to search for solutions to reduce the signal overlap. There's one possibility going to high fields, and there's also the possibility of going to high spinning speeds. So here is, again, paclitaxel now with the correct sum formula, and you see we have 51 protons. And because in the crystalline state, we have Z prime 2, it means we have overall 102 different proton environments that we would get signals from. And you see here the proton spectra of the crystalline material recorded between 40 and 100 kilohertz, and you can nicely see the increase of the resolution with increasing MIS frequency, which works really nice for this example. On the other hand, you see here the amorphous sample. So this is only the drug itself being amorphous. And you can see here that between 60 and 70 kilohertz, there's already barely an increase in the resolution. There are, of course, other benefits from fast MIS, but in the resolution, there's no increase, meaning that the intrinsic disorder of the sample prevents the resolution from increasing further because we have a distribution of similar but not identical environments. So fast MIS will probably be nice, but not the solution to all our problems. And then we had a second problem. We recorded data for the pure polymer, and we had experience with this blue polymer here in the bottom. And we could observe the carbon NMR spectrum. Every signal nice, we could assign them. And then we recorded another spectrum of this very, very similar, just one CH2 unit more in the side chain. And you can see where's the amide functionality, for example, why is everything so broad? And it took us a moment to trace this to the glass transition temperature of the polymer, also another property that we have to have in our minds, which is 50 degrees Celsius for the lower polymer and more than 60 degrees Celsius for the upper polymer. And if you think of the measurement temperature at 20 kilohertz where we did the recordings, then we have room temperature plus calibrated 27 due to friction and heating, and then we are roughly around or above the glass transition temperature of the lower polymer, but we are below the glass transition temperature of this other polymer. And when we realized this, we could of course solve this easily by just heating this polymer once over the glass transition temperature, and then you can see here the NMR spectra of the polymer above the TG, above the glass transition temperature. Now also the signal of the amides are here, and the other ones are better resolved. But still, if you look at the second spectrum, there's a lot of signal overlap between the polymer and the drug molecule, and also the signals are really broad. So we need additional input to help us improve and understand which of the effects stems from which part of our sample. And now I would like to come back to this relaxation discussion that we had in the beginning, and would like to introduce you to domain-edited NMR spectra. So this is a very nice work of Jiri Bruce and his group. So there's a sample which consists of crystalline domains, some also amorphous, and then crystalline molecules being included into this matrix. And you can see here that based on the T1 relaxation, you can discriminate between the two components, the orange blocks and the matrix. And then you can choose a specific condition that benefits so that you can simplify the spectra by making or taking advantage of different relaxation behavior. Unfortunately, as in our case, both species are amorphous. There's no large difference that we could observe. And this means that we couldn't go along this route with the relaxation behavior, but instead we took a second route and we used the fact that we have heteronuclei in our molecules. And in particular, we used 1415N, and because we have it only once in the drug and at specific sites in the polymer. The problem was that for the individual molecules, the chemical shifts in the 15N barely differed, so they were all around minus 250, which is why we went to 14N as a nucleus, and we made use of the second order quadrupolar shift to have a larger spread over this 14N shift region. And you can see here on this slide the individual spectrum of paclitaxel in crystalline form and of the polymer, which were our pre-experiments. And they were telling us, okay, great, we have something at roughly minus 20 ppm and something at plus 70 ppm. So both of the components are far enough from each other so that we can nicely trace effects that happen to the individual components. And then we measured this HMQC of the formulation, and we could see, okay, there are contacts and there are two different traces here at minus and there at plus. So we thought, great, and started to discuss and interpret. And then we realized, wait a moment, if we have this low paclitaxel loading, it means that only roughly 2% of all nitrogen atoms in our whole sample belong to the drug molecule, while the 98% belongs to the polymer because of all the repeating units. And this means that both of the signals that we observe in the NMR spectrum actually result from the polymer, and we only see the paclitaxel through the contacts that we have here by the polymer, but not in the nitrogen itself. And once we realized this, then we could analyze and interpret this, and we found, for example, interactions of the polymer with the nitrogen and oxygen polar groups of paclitaxel, and also could see that the nitrogen of the polymer gets more symmetric, so it's more like a tetrahedral environment rather than the initial asymmetric nitrogen environment. And this leaves me to summarize. After a short introduction, I've showed you 4 examples of learnings that I had when dealing with polymer samples that I hope you found useful. And as an outlook, I want to show you that the learning will fortunately never end, because I got excited by some of the work that is currently being done on targeted delivery, because the idea is really cool to have a targeting unit and then deliver a drug only to where it's necessary. And then I was fortunate to get samples from the Lammers group in Aachen, and also one of my students was willing to take this project on board, and we measured polymeric micelles with and without biotin at the outside. And you can see the spectra here. So in general, but what there is not is there's no signal whatsoever of these decorating units from the outside, because again, they are only at the outer parts, and you have many, many repeating units of the polymer, which means that we only observe in the spectra the polymer. And this is where it's the ongoing learning that I'm looking forward to. And with that, I have to really, really thank my team, which make it a pleasure every single day to come to work, and which motivate me every day to do the job. And thank you for your very kind attention. And I'm very much looking forward to the questions that you might have. Thank you.
Thank you so much for the great talk. Yeah, so questions. For the gelation, when you showed the gelation of the polymers after adding the drugs, can you track this? Like, is it sudden, or like, could you track it with the solid state NMR? Does it take a time to make hydrogels, or like it's very fast?
When adding the drug, I think it's not a highly structured hydrogel. So in that case, I think it's more that the particles stick together and then precipitate. So it's something that is rather fast, and that is also always observed when the polymer chemists prepare the formulations in solution, and then they usually filter or centrifuge the parts that are completely dissolved from the parts that have precipitated. But this is something that we observe regularly, and I agree that, for example, with the work that Julia Mollica is doing, or the group in Cardiff, with the crystallization, maybe there's also a possibility of doing a similar analysis with such amorphous compound. Yeah, would be thinkable. Yeah, thank you for the idea.
Okay, cool. So we have another question for the bio from Yongsho. So, for the biorelevant media, the I don't know, passive and fisive,
Yeah.
can DOSY NMR differentiate and quantify the micelles and monomers of surfactants in the solution?
So what we can do is that we can calculate the sizes of the micelles and can compare them with what we observe from cryotem, for example. So if we make a size distribution over all the cryotem images that we have and compare it to the average size that we calculate for the micelles, they are in the same dimension. So we can transfer it, and we can also, for example, follow a trend in increasing size or decreasing size. What is difficult is we are currently saying that all the long alkyl chains, we can't separate sufficiently between them. So for some species, there are individual functional groups like double bonds, for example, in the backbones, and then they are nicely separated and we can analyze them, or the methyl groups for the taurocolate. But that's a hurdle that many of the signals are strongly overlapping, let's say, in the middle region of the spectrum. I hope that answers. Otherwise, just add a second question to that.
Okay, cool. I have another question for the polymers. What controls the interaction? Is it like hydrophobicity, hydrophilicity, the main factor, or there is an actual binding with the drugs?
So for the polymer self-assembly themselves, the main driving force would be the ratio of the hydrophilic and hydrophobic segments. And then as soon as the drug molecule comes in, there are additional interactions. So for example, for the drug molecules, we can clearly, for example, see experimental proof and also combine it with calculations that, for example, OH groups in, now my light is switching off, that OH groups in the drug molecule would, for example, interact with the amides in the polymer. For the hydrogels, it gets more complicated because the hydrogels form these worm-like structures, but there's no polar group that could facilitate this. And so there are lots of different interactions involving the carbonyls, for example, and the pi electrons discussed. But I would say that we have indications there, but it's not finally clarified. So this is still ongoing.
Okay, cool. So I didn't see is there a question? Oh yeah, we have a question from Blake. You showed the hydrogel forming polymers in transition from a hydrogel state at low temperature to a micelle state at high temperature.
Yeah.
Does the hydrogel state reform upon cooling?
Yeah. So you can do repeated heating and cooling circles, and it would always reversibly form. So that's no problem.
Okay, so I don't see. Oh yeah, no, there's another question. Can DOSY based from Mushi, can DOSY based methods be useful to investigate drug release? For example, a micelle broke and the content entered the solution.
Yeah. I think what makes this complicated is that so the release is certainly an important factor that we need to take into consideration. And we can, for example, see also in the DOSY, in these comparisons with different concentration profiles that I've shown, that at some point the molecules must leave the micelle and be associated with the other parts of the mixture. But the whole dynamics of the process, we are currently not able to capture. So I think that the kinetic part of this system is something that we are at the moment not valuing enough. So I think this would be interesting to go into more detail. So you're right with your questions. And I'm not sure. So about the DOSY, there's sure more potential, but what we also explored recently or started to explore was to have barriers in our sample and then combine the aspect of diffusion and the aspect of having a real physical barrier so that we can separate the different states better. So maybe a combination of this would probably give us more insight than just DOSY alone.
Okay. And last question, from someone with little experience with DOSY. What are the important differences between a diffusion optimized probe and a conventional solution NMR probe?
Okay, so there are more DOSY probes out there, but the one that we are operating with, we have one of the standard solution probes with a gradient with which we can do the normal DOSY measurements. And then we have a specific diffusion probe, which has a specific gradient, which is stronger than the one in the normal probe. And this means that we can monitor processes of diffusion that are much smaller than the ones that we can observe with a small gradient, because with the normal gradient, we would need to push with all the parameters that we have the system to the limit. And once we've reached the limit conditions, then we cannot improve further. But if we switch to the stronger gradient, then we can, for example, have observe a stronger effect in a shorter diffusion time. And then, for example, even the network wobbling in a hydrogel would be something in this order of magnitude that you could still see or get information on. So you're going to a lot slower processes with a specific diffusion probe. And I know that the current ones also have more, the one that we have only has protons and carbons, and the modern ones also have broadband, which makes it really attractive for a very broad range of materials as well.